Hello friends, today we'll learn about micropropagation. Today this video is the second video of micropropagation. Now let's start with genetic variability. When micropropagation is carried out through shoot tip cultures, genetic variability is very low. However, use of adventitious shoots is often associated with pronounced genetic variability. Now, let's know about vitrification. During the course of repeated in vitro shoot multiplication, the cultures exhibit water-soaked or almost translucent leaves. Such shoots cannot grow and even may die. This phenomenon is referred to as vitrification. Vitrification may be prevented by increasing the agar concentration in the medium. However, increased agar concentration reduces the growth rate of tissues. Now let's know about cost factor. For some propagation new techniques, extensive equipment, sophisticated facilities and trained manpower are needed. This limits its use. Now let's know about the production of disease-free plants. How we can produce disease-free plants. Many plant species are infected with pathogens, viruses, bacteria, fungi, mycoplasma, and nematodes that cause systematic diseases. Although these diseases do not always result in the death of plants, they reduce the quality and yield of plants. The plants infected with bacteria and fungi frequently respond to chemical treatment by bactericides and fungicides. However, it is very difficult to cure the virus infected plants. Further, viral diseases are easily transferred in seed propagated as well as vegetative propagated plant species. Plant breeders are always interested to develop disease free plants, particularly viral disease free plants. These have become a reality through tissue cultures. Now let's know about the apical meristems with low concentration of viruses. In general, the apical meristems of the pathogen infected and diseases harboring plants are either free or carry a low concentration of viruses for some regions. Let's know about the reasons. First point is absence of vesicular tissue in the meristems through which viruses readily move in the plant body. Next point is rapidly dividing meristematic cells with high metabolic activity do not allow viruses to multiply. Virus replication. Next point is virus replication is inhibited by a high concentration of endogenous oxygen in shoot apexes. Tissue culture techniques employing meristem tips are successfully used for the production of disease-free plants caused by several pathogens, viruses, bacteria, fungi, mycoplasmas. Now let's know about the methods to eliminate viruses in plants. In general, plants are infected with many viruses. The nature of some of them may be unknown. The usage virus-free plant implies that the given plant is free from all viruses, although this may not be always true. The commonly used methods for virus elimination in plants. Now we will know. Heat treatment of plant, meristem tip culture, chemical treatment of media, and other in vitro methods. Now, first point is heat treatment method. That means thermotherapy of plants. In the earliest days, before the advent of meristem cultures, in vivo eradication of viruses from plants was achieved by heat treatment of whole plants. The underlying principle is that many viruses in plant tissues are either partially or completely inactivated at a higher temperature with minimal injury to the host plant. Thermotherapy, that means a temperature of 35 to 40 degrees centigrade, was carried out by using hot water or hot air for elimination viruses from growing shoots and buds. There are two limitations of viral elimination by heat treatment. First point is most of the viruses are not sensitive to heat treatment. Second point is many plant species do not survive after thermotherapy. With the with these disadvantages, heat treatment has not become popular for virus elimination. Now let's know about the meristem tip culture. For viral elimination, the size of the meristem used in cultures is very critical. This is due to the fact that most of the viruses exist by establishing a gradient in plant tissues. In general, the regeneration of virus-free plants through cultures is inversely proportional to the size of meristem used. The meristem tip explant used for viral elimination cultures are too small. A stereoscopic microscope is usually employed for this purpose. Meristem tip cultures are influenced by some factors. Let's know about the factors. Physiological conditions of the explant actively growing buds are more effective. Next point is thermotherapy prior to meristem culture. For certain plants, heat treatment is first given and then the meristem tip culture are isolated and cultured. Culture medium. MS medium with low concentration of auxins and cytokinins is ideal. Now let's know about the chemical treatment of media. Some workers have attempted to eradicate viruses from infected plants by chemical treatment of the tissue culture media. The commonly used chemicals are growth substances like growth hormones like cytokinins and antimetabolites used like thiouracil, acetyl salicylic acid. There are, however, conflicting reports on the elimination viruses by chemical treatment of the media. For instance, addition of cytokinin suppressed the multiplication of certain viruses while for some other viruses it actually stimulates. Now let's know about some in vitro methods. Besides meristem tip culture, other in vitro methods are also used for raising virus-free plants. In this regard, callus cultures have been successful to some extent. The callus derived from the infected tissue does not carry the pathogens throughout the cells. In fact, the uneven distribution of tobacco mosaic virus (TMV) in short TMV in tobacco leaves was exploited to develop virus-free plants of tobacco. Now, somatic cell hybridization, gene transformation, and somaclonal variations also useful to raise disease-free plants. Let's know about the elimination of pathogens other than viruses. Besides the elimination of viruses, meristem tip cultures and callus cultures are also useful for eradication bacteria, fungi, and mycoplasmas. And let's know about some examples. The fungus Fusarium roseum has been successfully eliminated through meristem cultures from carnation plants. Certain bacteria like Pseudomonas carophili, Pectobacterium partheni are eradicated from carnation plants by using meristem cultures. Now let's know about the merits and demerits of disease-free plant production. Among the culture techniques, meristem tip culture is the most reliable method for virus and other pathogen elimination. This, however, requires good knowledge of plant pathology and tissue culture. Virus-free plants exhibit increased growth and vigor of plants, higher yield, example potato, increased flower size, chrysanthemum, improved rooting of stem cuttings, example pelargonium. Virus-free plants are more susceptible to the same virus when exposed again. This is the major limitation. Reinfection of disease-free plants can be minimized with good knowledge of greenhouse maintenance. Now let's know about the embryo culture. Embryo culture deals with the sterile isolation in vitro growth of the mature and immature embryo with an ultimate objective of obtaining a viable plant. Conventionally, the term embryo culture refers to the sexually produced zygotic embryo culture. This is different from the somatic embryogenesis, and there are two types of embryo cultures, that is mature embryo culture and immature embryo culture. Now, first, let's know about the mature embryo culture. Mature embryos are isolated from ripe seeds and cultured in vitro. Mature embryo cultures are carried out in some conditions. First condition is when the embryos remain dormant for long periods. Second point is low survival of embryos in vivo. Third point is to avoid inhibition in the seed for germination. Fourth point is for converting sterile seeds to viable seedlings. Seed dormancy in plant species is a common occurrence. This may be due to chemical inhibitors or mechanical resistance exerted by the structures covering the embryo. Seed dormancy can be successfully bypassed by culturing the embryos in vitro. Embryo culture is relatively easy as they can be grown on a simple inorganic medium supplemented with energy source. This is possible since the mature embryos excised from the developing seeds are autotrophic in nature. Now let's know about embryo rescue. Embryo rescue involves the culture of immature embryos to rescue them from unripe or hybrid seeds which fail to germinate. This approach is very useful to avoid embryo abortion and produce a viable plant. Wild hybridization involving crossing of two different species of plants from the same genus or different genera often results in failure. This is mainly because the normal development of zygote and seeds is hindered due to genetic barriers. Consequently, hybrid endosperm fails to develop, leading the abortion of a hybrid embryo. The endosperm may also produce toxins that ultimately kill the embryo. In the normal circumstances, endosperm first develops and supports embryo development nutritionally. Thus, majority of embryo abortions are due to fail in endosperm development. Embryo abortion can be avoided by isolating and culturing the hybrid embryos prior to abortion. The most important application of embryo rescue is the production of interspecific and intergenic hybrids from wild plant species. Now let's know about the culture techniques for embryo rescue. The isolation of immature embryos often poses some difficulties. The aseptically isolated embryos can be grown in a suitable medium under optimal conditions. In general, a complex nutrient medium is required for culture methods involving embryo rescue. For adequate nutritional support of immature embryos, embryo endosperm transplant is used. Let's know about the embryo endosperm transplant. The endosperm transplant technique used for culturing immature embryos. The hybrid embryo from the ovule in which endosperm development has failed is taken out by excision. Another normally developed ovule which with endosperm enclosing an embryo is chosen. This ovule is dissected and normal embryo is pressed out. This leaves a normal endosperm with an exit hole. Now the hybrid embryo can be inserted into the normal endosperm through exit hole. This results in embryo endosperm transplant which can be cultured in a suitable medium. By using embryo endosperm transplant, Many interspecific and intergenic plants have been raised. Example, hybrid plants of legumes. Now let's know about the nutritional requirements of embryo cultures. There are two phases in the embryo development and nutritional requirement varies accordingly. So let's know about heterotrophic phase. First phase is heterotrophic phase. This is an early phase and the embryo is mostly dependent on the endosperm and maternal tissues for nutrient supply. Now let's second point. Second phase is autotrophic phase. This phase is characterized by the metabolic capability of the embryo to synthesize substances required for its growth, which slowly makes it independent. The critical stage is the intervening phase between the heterotrophic and autotrophic phases. The nutrient supply is highly variable at this phase, which mostly depends on the plant species. In general, the composition of the medium for culturing immature embryos is more complex than that required by mature embryos, which can grow on a simple inorganic medium. Further, the transfer of embryos from one medium to another is frequently needed in order to achieve full development of embryos. Now, let's know about the composition of medium. Let's know about some. Let's know about composition of medium. First is inorganic constituent, that is MS medium, B5 medium, White's media are adequate. Sucrose is most commonly used for energy source. Ammonium nitrate is the preferred source of nitrogen. Casein hydrolysate, rich in various amino acids, is frequently used. Certain natural plant extracts with embryo factor promote embryo cultures. Example, liquid endosperm of coconut milk. The embryo factor is believed to supply certain amino acids, sugars, growth regulators, etc. In general, growth regulators are not required as they induce callus formation. Embryos grow well in the pH range of 5.5 to 5 pH to 7.5 pH. Uh, An incubation temperature of 24 to 26 degrees centigrade is ideal. Better growth of embryos is observed in darkness, which are then transferred to light for germination. During the culture conditions, embryos are grown into plantlets and then transferred to sterile soil for full full-fledged growth to maturity. Now let's know about the applications of embryo cultures. First point is prevention of embryo abortion. Incompatibility barriers in interspecific and intergeneric hybridization programs leading to embryo abortion can be successfully overcome by embryo rescue. In fact, many distant hybrids have been embryo uh, obtained through rescue techniques. Now, second point is overcoming seed dormancy. Seed dormancy is caused by several factors: endogenous inhibitors, embryo maturity, specific light and temperature requirements, dry storage requirements, etc. Further, in some plants, the natural period of seed dormancy itself is too long. Embryo culture is successfully applied to overcome seed dormancy and to produce viable seedlings in these plant species. Next point is shortening of breeding cycle. Some of the plants in their natural state have long breeding cycle. This is mostly due to seed dormancy attributed to seed coat and endosperm. The embryos can be excised and cultured in vitro to develop into plants within a short period. For instance, hollies, a Christmas decoration plant, can be grown in 2 to 3 weeks time through embryo cultures, in contrast to 3 years period required through seed germination. Now, let's know about production of haploids. Embryo culture has been successfully used for the production of haploid plants. Example barley. It's or monoploid. Now let's know about the next point. Overcoming seed sterility. Certain plant species produce sterile seeds that do not germinate. Example early ripening varieties of cherry, apricot, plum. Seed sterility is mostly associated with incomplete embryo development, which leads to the death of germinating embryo. Using embryo cultures, it is possible to raise seedlings from sterile seeds of early ripening fruits. Example apricot, plum. Next point is clonal propagation. Embryos are ideally suited for in vitro clonal propagation. This is due to the fact that embryos are juvenile in nature with high regenerative potential. Further, it is possible to induce organogenesis and somatic embryogenesis from embryonic tissues. Thank you, students. Any questions regarding this? You can ask me in the comment box and subscribe my channel. And if you want more videos about biotechnological topics, you can subscribe my channel to know more. Thank you.